Hello everyone, let us study a very interesting topic in molecular biology, that is packaging of DNA helix. To start with, length of DNA double helix is calculated by the formula total number of base pairs present in DNA multiplied by distance between the two base pairs. If we take for example a mammalian cell which represents a typical eukaryotic cell, in particular human cell, the length of DNA is 6.6 into 10 to the power of 9 base pairs, and it should be multiplied by the distance, that is 0.34 into 10 to the power of minus 9 meter. Then we will get the length of approximately 2.2 meter. We know that since human cells are eukaryotic, and in all eukaryotic cells, whether it may be mammalian or any other cell, DNA is present in a structure called the nucleus. If we take the length of DNA as 2.2 meter in human cell, and it will be present inside a very small structure called nucleus, which dimension is 10 raised to minus 6 meter. And how it is possible? Let us study that later. To understand this topic, first let us study in case of prokaryotic cell. For example, Escherichia coli, the length of DNA is 1.36 millimeter. But in prokaryotic cell, there is no nucleus present. And we think the DNA may be scattered there. But in prokaryotic cell also, DNA is not scattered here and there. If we take this as a typical prokaryotic cell, the DNA is held together at some places by a proteins, and it will result in the formation of a structure called nucleoid. Here in the term nucleoid, oid refers to like, means nucleus like structure present in prokaryotic cell is called nucleoid. And here the loops of DNA are formed when they are associated with the proteins, which results in a formation of single chromosome that you have studied in first year chapter, that is biological classification. Now in case of eukaryotic cells, the organization of the DNA is much complex. Here there is presence of a proteins called histone proteins, and these proteins are positively charged, and this charge is depending upon the abundance of amino acid residues with the side chain. If we imagine a protein, it will have a side chain which is having some charge, so that will give a particular charge. In case of histone proteins, the abundance of lysine and arginine will give its nature, that is positive charge, which are basic amino acids found in histone. There are 5 types of histone proteins: H1, H2A, H2B, H3 and H4. 8 molecules of particularly H2A, H2B, H3 and H4 will result in formation of a organized unit called a histone octamer. Here, if I am writing here, there are 2 molecules of H2A, 2 molecules of H2B, 2 molecules of H3 and H4. All together, it will forms a one unit that is called histone octamer. Since the proteins are positively charged, the histone octamer also got a positive charge. We know that DNA is negatively charged molecule because of the negatively charged phosphate group, of course. The negatively charged DNA will coil around the positively charged histone octamer to form a structure called nucleosome. And this nucleosome DNA is supported by the one more subunit of histone, that is called H1 histone. Likewise, there is formation of many number of nucleosomes when the DNA, total length of DNA, when it coil around the histone octamer in eukaryotic cell. This is very, very important. And these many nucleosomes together will form a structure called chromatin, and this chromatin can be seen during the interphase when the cell is present at rest. And when we observe this chromatin under electron microscope, it just appears like a beads on string. In your NCERT, the diagram of nucleosome is given, which represents a core of histone proteins and a DNA which is coiled around it, and the DNA which is present between the nucleosomes is supported by a subunit that is called H1 histone. And this is very important for 2 mark for theory exams, diagram, and for competitive they may ask it for diagram based question. Now, when one nucleosome is connected with another nucleosome, there is short length of DNA present, that is called linker DNA, and that linker DNA is supported by the structure called histone proteins, H1 histone. Now, many such nucleosomes are arranged to form a beads on string, and this beads on string structure is packaged to form a structure called chromatin fiber. This is the next level of organization. Basic level of organization is nucleosome. So nucleosomes will give rise to beads on string structure. Beads on string structure will be packaged to form a chromatin fibers, and chromatin fibers are further coiled by the process called condensation during the metaphase of cell division. You have to remember the nucleosomes or chromatin is seen during interphase. But during metaphase, the higher level of condensation will take place, which results in the formation of a structure, a unit that is called chromosome. In humans, there is, as we know, there is the formation of 46 number of chromosomes. So now, when we uncoil a chromosome, what we get? First, we will get a chromatin fiber. Further uncoiling will give us chromatin, and further uncoiling will give us a nucleosome, and further uncoiling of nucleosome will give us a histone proteins which is separated from the DNA, negatively charged DNA. Now, when we observe the higher level of condensation or complexity of DNA packaging, first DNA will coil around positively charged histone proteins to form nucleosome, and nucleosomes will be organized to form a chromatin, that is during interphase of cell division, that is the cell is at rest, and it will appears like a beads on string structure when we observe under electron microscope. And this beads on string structure will further undergo further condensation, that is called chromatin fibers, and these chromatin fibers will further get associated with non-histone chromosomal proteins, and which will result in formation of a structure called chromosome, and this is called packaging at higher level. Lower level will be nucleosome and higher level will be the chromosome. To form a chromosome, there is a requirement of specialized proteins apart from histone. They are called non-histone chromosomal proteins. You have to remember the short form and full form. NHC refers to non-histone chromosomal proteins. When we see a nucleus under electron microscope or any simple microscope, actually it is not visible under a compound microscope. So here within nucleus, the chromatin is highly coiled or loosely packed. So highly packaged or loosely packaged. Depending upon that, there are two regions found in nucleus, that is called euchromatin and heterochromatin. Difference between these two is very important, both for competitive as well for theory. Euchromatin are the regions of chromatin that are loosely packed, but heterochromatins are densely packed. Euchromatins will stain light after staining procedure, whereas heterochromatins will stain dark. Whereas euchromatins are transcriptionally active, that means they undergo the process called transcription to form a mRNA, whereas heterochromatins are transcriptionally inactive because of their packaging. The concept you will study still later. Thank you.